So our next speaker is Gerard Wong, who will talk about polyelectrolyte physics meets innate immunity and autoimmunity.
Okay. So first of all, I want to thank the organizers for inviting me. This talk will be a little bit different. I'm going to talk about polyelectrolyte physics in the context of innate immunity and autoimmunity. And I'm going to start with two diseases, lupus and psoriasis. And in both of these diseases, it comes from a defect in immune tolerance to self-nucleic acids. If you look at lupus, it's a systemic disease, and right now there are essentially limited treatment options, and these treatment options consist of essentially using anti-inflammatory drugs to knock down the immune system. If you look at psoriasis, this is an autoimmune skin disease, and in this case also there isn't really a cure, and it's also treated with systemic immunosuppressants. The newest treatment options will include, mechanisms to knock down individual interleukin signals, but even then, it essentially is an immunosuppressant. And when you suppress the immune system, you're going to have significant side effects. Now, in both of these diseases, the disease essentially comes from the hyperactivation of toll-like receptors by self-nucleic acids. Toll-like receptors are part of our immune system's technology for detecting nucleic acids. And in this case, both self-DNA and self-RNA are involved. Now, if you think about how our bodies detect nucleic acids, we use various so-called pathogen-associated molecular patterns. And these patterns are recognized by various sensors. So you have endosomal sensors like the toll-like receptors. So in the case of, double-stranded RNA is detected with toll-like receptor 3, single-stranded RNA is detected by toll 7, 8, and double-stranded DNA is detected by toll 9. You also have cytoplasmic sensors like the STING pathway and so forth, and these I'm not going to talk about today, but they're actually quite hot topics in immunology right now. It was found a few years ago that there is an antimicrobial peptide or innate immune peptide, LL37, found in humans that's hyperexpressed in psoriasis disease, for example, and it condenses DNA into complexes that wind up amplifying toll9. And this leads to highly amplified production of cytokines and leads to huge immune outcomes. So we want to look at this process a little bit more closely. LL37 looks like this. It's a bent alpha helix. It's cationic. It's got +6 charge. It's facially amphiphilic. And the aspect ratio is about 5:1. If you look at toll-like receptor 9, toll-like receptor 9 looks like two cationic paddles that grab onto DNA. And DNA, as you know, is highly anionic. These are three different views of how DNA binds to toll9. And so what do? We wanted to look at this process of immune activation a little bit more closely. How does the complex formation between LL37, this helical amphiphile, and double-stranded DNA wind up giving you this highly immunogenic structure? So we basically did some synchrotron X-ray diffraction experiments. And what we found was the following. We looked at a large family of these condensed structures. We looked at basically complexes formed between LL37 and DNA, but also between other cationic ligands and DNA. And what we did was we generated a plot that looks like the left panel here. What we're doing is we're looking at the immune response on the vertical scale versus the first peak position you get in X-rays, which, as you know, is roughly inversely proportional to the dominant periodicity in the system. And you see something very striking. You see that there is a sweet spot. There is like a periodicity that is preferred And when you're at this periodicity, your immune response skyrockets. If you're at a periodicity that's either smaller or larger, the immune response goes way down. And so we looked at this and we asked, like, what is so special about this periodicity? And the hypothesis we came up with was it actually matches up quite well with the steric size of toll-like receptor 9. So we thought maybe when you form a complex, and you form a complex at the right geometric spacing, it presents this multivalently to an array of close-packed toll-like receptors, and this will lead to an amplified outcome. So to test this idea, we first of all, we did a bunch of microscopy experiments that do double and triple co-localization. And to make a long story short, we found that immune cell activation really isn't due to differences in cell entry or endosomal access, because if you think about it, toll-like receptor 9 lives in the endosomes of these immune cells. So a reasonable hypothesis would be these different structures of these complexes have different levels of access to the endosome. And that turns out to not be the explanation. So if that's not the explanation, then what is the explanation? So we did this work together with Jure Dobnikar and Don Frankel. And the idea is the following. They did a coarse-grained computer simulation. They looked at what happens when you look at DNA lattices, two-dimensional DNA lattices, at different spacing that's being presented to an array of toll-like receptors, which is represented by this green piece that you see binding up top. And what they see is that as you change the inter-DNA spacing to a periodicity that matches up with toll-like receptor 9, what you get is a massive recruitment of toll-like receptor 9 to the binding region. You get a lot more, you change the free energy landscape of binding, so you get a lot more of toll-like receptor 9 going to the binding region. So this is very interesting because if you look at what's been happening in the immunity field in the last 20 years or so, you'll see that a lot of emphasis is placed on the idea of receptor clustering that gives you an immune amplification. So what we're seeing here is if DNA gets organized into a complex, not any complex, but a complex with the right geometric spacing and presentation, this is a kind of ligand clustering that can drive receptor clustering and give you a huge immune amplification. So we wanted to check the generality of this idea. Does it go beyond toll-like receptor 9? Does it go beyond LO37? So the first thing we did was we looked at amyloids, and you've all heard of amyloids. These are known to be cytotoxic to neurons. These are known to also be very pro-inflammatory. So we looked at two different kinds of amyloids. We looked at amyloids generated from DNA and lysozyme, amyloids of bacterial origin generated from the so-called curly fibers, which are bacterial amyloids found in biofilms. In this case, we used salmonella biofilms, and we looked at complexes formed between these curly fibers and DNA. In both of these cases, if you look at the X-ray scattering, you find that you get ordering of DNA into the periodicity that gives you this kind of immune activation. And this work we published last year. We wanted to look more broadly. I mean, does it have to be an alpha helix? Does it have to be an antimicrobial peptide? So we worked together with an immunology group at the University of Rome. And they're interested in a disease called scleroderma, or systemic sclerosis. This is the disease that Paul Klee died of, if you are a fan of modern art. In this case, it's known that there is a cationic chemokine, CXCL4. This is a mixed alpha helix beta sheet molecule, and it's known as a signaling molecule. So if you look at CXCL4, it's highly overexpressed in human patients' blood. And it was generally assumed that the only role that this plays is really the signaling role. But what we did when we looked at CXCL4 is that it actually also forms complexes with DNA, and it also organizes the DNA into this magic spacing that is highly pro-inflammatory. And in fact, if you look at the pathogenesis of systemic sclerosis, you find that it also acts through a Toll9 pathway that detects double-stranded DNA in the way that we have described. And so this is work that we are in the process of finishing up, where we combine human data with the X-ray diffraction and so forth. Now, you might say, okay, so far you only showed us examples based on Toll-like receptor 9. What about some of the other Toll-like receptors that detect other types of nucleic acids? So here we worked with Rich Gallo's group at UC San Diego. He's a dermatologist, immunologist who works on the skin disease psoriasis. And here we are interested in Toll-like receptor 3, which detects double-stranded RNA. And the interesting thing here, in terms of the generality of this picture, is we're now no longer looking at plasmacytoid dendritic cells, which is what we did before with the Toll9 story. We are using keratinocytes because this is, in many ways, very relevant to skin diseases. These are essentially skin cells. And instead of looking at an interferon alpha readout of your immune amplification, we're looking at the readout based on IL-6, which is a different cytokine signaling molecule. And what we see, if you look at the results, is exactly the same thing. We're finding that, again, if you look at the same kind of plot where you look at the increase in your immune signal as a function of your first peak position, you again have this kind of sweet spot. And this sweet spot, again, corresponds very closely to the expected size of Toll-like receptor 3. So we looked at this and we thought, okay, maybe we're on the right track. But so far you might say, okay, you've only shown us data that shows immune amplification. Can it modulate the immune system in any other way? So we started working together with a group at USC, and they are very interested in a antimicrobial peptide from the old world monkeys. This is interesting because this so-called Theta defensin from the rhesus monkey, this is a circularized peptides with an array of disulfide bridges. What you wind up with is a molecule that has a very high cationic charge, and it is also very small and very flat. And when they look at the function of this molecule, they found that not only can it punch holes in bacterial membranes, it's also known to be anti-inflammatory. It seems to suppress inflammatory cytokines in sepsis models. It also seems to inhibit the so-called NF-kappa B pathway, which is the immune cascade that is related to various Toll-like receptors, including Toll1, 2, 4, and 9. And so we decided to look at this more closely. And when we looked at the X-ray diffraction picture from Theta defensin, what we find is that it packs the DNA together so closely that it puts it way, way out of the pro-inflammatory range, as you can see from the picture on the left. But now it gives you actually a very interesting idea, because now what you can think of is a kind of competitive binding between these Theta defensins and your pro-inflammatory peptides. So we did an experiment where we looked at a ternary system with DNA, the pro-inflammatory LL37 peptides, and the anti-inflammatory RTD1s, which are the Theta defensins. And we find that they will outcompete LL37 and make their own spacing, which is not pro-inflammatory. It puts a spacing at the green arrow that we've shown. So this is interesting because if you now take this to a mouse model, and this is what Soustedt and Wallet did, what we find is this is an example of a healthy mouse. And it's running around, it's curious, it's doing various things. And then the next segment of the movie will show what happens to a mouse with rheumatoid arthritis. This is an autoimmune disease, as you know, and it's related to the Toll3 pathway as well as a couple of others. And then they're going to show the third part of the movie, which is going to be what happens when the arthritic mouse gets exposed to the Theta defensins and it's able to walk again. And no one really knows why this is. And I think the picture that we show actually gives a a kind of interesting hypothesis for what might be causing these types of anti-inflammatory effects, that it's outcompeting some of the pro-inflammatory peptides that give you these kind of pathological immune outcomes. So, okay, so far I haven't said anything about structures. I just showed you what the effects are. I showed you that the internucleic acid spacing seems to be important. I didn't talk about this next part, but it turns out the number of repeating unit cells also seems to be important, as our data with double-stranded RNA showed. But the general question I want to ask is, how do these helical peptide amphiphiles, like LL37, wind up self-assembling with DNA to form these immunogenic, essentially nanocrystals? And this is a problem where you have two different principles of ordering, electrostatic and amphiphilic self-assembly, which is something you've heard about in this conference. There's like great work that you heard about in this morning session. But if you think about amphiphilic self-assembly with simple shapes that you've heard of in soft matter, obvious cases to think about would be lipid amphiphiles and the fact that you can make micelles and have the CMC. You can also think about the so-called Janus particles and chemically patchy particles that people have been studying in the last 10 years. The nice work from Steve Granick and Sharon Glotzer, Eric Bloughton, Andy Ferguson, many others. What if you use these non-canonical shapes, right? These weird, low symmetry, bent alpha helices that are facially amphiphilic, that have like ridges and depressions and so forth. How do they come together? And what happens when you throw in electrostatics into this picture? And the basic question I'm asking is, when you have a peptide that's helical, cationic, and amphiphilic, what are the physical chemical requirements in order for these alpha helices to be immunogenic in the mechanisms that I have just described? And you can see this is an important question because alpha helices are pretty much everywhere, and it would be important to know when they can actually supercharge your immune system and lead to unforeseen consequences. So if you look closely at LL37 has got +6 charge. If you look at it on the helical wheel, kind of endwise, you'll see that the hydrophobic phase, sub-10, is about 120 degrees. It's got a little bit of a bent via a glycine. If you look at DNA, everybody in this room knows that DNA has -2 charges every 0.34 nanometers, and it's about 2 nanometers in diameter. But you have a puzzle. If you look at the inter-DNA spacing that we get from the small angle synchrotron and X-ray scattering, this is like way too big for a single LL37 molecule to bridge. And the answer turns out to be the following, that LL37, in the presence of oppositely charged DNA, will actually form a fibril. This is work that we did together with Peng-Yu Ren's group at the University of Texas at Austin. And the idea is that the LL37, which is this bent alpha helix, forms a kind of superhelix where the core of this superhelical fibril is hydrophobic, and the outside is cationic so that it can interact efficiently with the DNA. So it co-assembles into this columnar square lattice. If you look at the cross-section, you see 4 LL37 molecules in the cross-section, and this is the structure that winds up maximally activating. If you look at another alpha helical AMP, if you look at melatonin, this is from bee sting venom, again, you get something that's very similar. And again, you get this sort of like 3-dimensional tetragonal lattice with the same kind of fibril where you have this kind of 4-fold coordination with the hydrophobic core. Can we try to understand this? So from electrostatics, you can kind of imagine that, well, if this is driven by the entropy gain of counterion release between the DNA and the oppositely charged cationic fibril made from these antimicrobial peptides, you would imagine that you would get the strongest interactions when these charge densities are equal and opposite. And if you look at the fact that we have this 4-fold coordination, this actually gives you exactly that kind of charge density matching. You have about -20 charges in the DNA every 3.4 nanometers, and you have about the same amount of positive charges when it goes into this 4-fold coordination. What about hydrophobicity? For hydrophobicity, why is this protofibril 4 alpha helices thick? And if you think about how much the individual helices have, I mean, what sectors are subtended by these hydrophobic patches, you can kind of imagine that in order to stabilize the hydrophobic core, you need a certain minimum amount of hydrophobicity. And in this sort of like simple cartoony picture, we were guessing that if it gets below about 90 degrees, it would have trouble maintaining this kind of hydrophobic core in order to give you the good electrostatics. So we looked at yet another antimicrobial peptide besides LL37 and melatonin. This is Gouffran. This is something that has about an 80-degree hydrophobic phase. What we get is, if we look at the X-ray scattering, you get a peak at the right spacing, but you lose the lattice. It becomes disordered, which we imagine happening when you have a system that flickers between different coordination states because it can't really stabilize these types of fibro cores. So this seems to make sense, but how does this now dovetail with the immune experiments? The immune experiments actually follow the hypothesis fairly well. What we find is that LL37 led to high amplification levels, which makes sense. Melatonin, we also expected to lead to high amplification levels, but they wound up killing immune cells because they are so good at permeating their membranes. And as expected, if you look at Gouffran, it led to low immune amplification levels. So we were a little bit bothered by the melatonin case because we were expecting it to have a very high response. But can you now take melatonin and re-engineer it so that it has less membrane activity? And this is something that can be done. We've actually pursued a machine learning algorithm that allowed us to assay the degree of membrane activity, and we can also construct these mutants based on what the literature has indicated. And we can make mutants of melatonin that don't permeate the membrane quite as well, and we actually can recover the high levels of Toe9 activity even for melatonin. And so this is interesting because now it shows that we can take an alpha helix and we can engineer in like immunomodulatory activity in a way that seems deterministic. And so now I'm in a session where I'm sandwiched by two nucleosome experts. So we decided maybe we'll end the story with an example that has to do with nucleosomes. So I want to talk about three different kinds of cell death. So there is apoptosis, which is the cleanest form of cell death you can have. There's necrotic cell death, which is a very messy kind of cell death that involves the spilling of chromatin into the extracellular space and also the release of calcium ions. The picture we have in the bottom are basically histology slides that shows what happens after a heart attack. You can see that there is like a huge infiltration of neutrophils that comes from this kind of autoimmune reaction. Why is it that our dead cells will elicit this immune response, right? And it turns out there's a phenomenon called nets formation or netosis. This is a kind of controlled neutrophil cell death via a kind of self-destruct sequence. And what immune cells, what neutrophils specifically do, is they basically extrude a web of chromatin that's complexed with various proteins that winds up forming a kind of trap for pathogens. And this is, a very complicated polyelectrolyte soup that is, non-equilibrium and heterogeneous. It includes nucleosome core particles. It includes some of the antimicrobial peptides I had talked about, like LL37, human beta defensin 3, and lysozymes, and so forth. It includes also DNA and nucleases to cut up the DNA and pad enzymes to turn arginines into citrulline and wind up decreasing charge of these various cationic components and so forth. So what happens there? The reason we're interested in this is because these so-called NETs components that I just listed are important in all kinds of autoimmune phenomena, including lupus, including psoriasis, and others. And right now, the mechanism for the immune activation is not understood. People do know, however, that NCPs activate inflammation through the Toe9 pathway. So we wanted to see what happens. And as you heard from the previous talk, there is a propensity of of nucleosomes to stack. And this is work that we did together with Lars Nordenskjold here at NTU and his team. And what we did was we took these nucleosome core particles, we added a little bit of calcium, the amount that is consistent with what you get physiologically during nets formation. And we find that these NCPs stack together to form a column. The thing that's interesting is when it forms a column like this, in order for the DNA to remain isoelectric with the charge on the column, they will change the DNA helical pitch slightly to accommodate, because now the boundary conditions are different. It's no longer wrapping about 1.8 times around each NCP. It can actually relax that condition slightly. And what we find is if you look at the bar graph up top, we find that NCPs by themselves are not immunogenic, but you add a little bit of calcium so that they form these columns. Now the interferon alpha response in plasmacytoid dendritic cells goes through the roof. And if you now knock down the TOL9 pathway using chloroquine, it goes back down to 0. So we think one of the reasons that these pro-inflammation structures form has to do with the polyelectrolyte physics of what happens when you wrap DNA around like a cylinder instead of one of these hockey pucks. And what happens if you were to add in LL37, which is known to also be found in these so-called nets. We find that these columns actually will order into a columnar hexagonal array where you again get this kind of multivalent presentation of DNA onto toll-like receptors that will give you this kind of amplification. And again, when we look at NCPs only, NCPs plus LL37, and the whole system knocked down using chloroquine, you see the characteristic low, high, low profile. So we're in the process of kind of thinking through this with an immunologist, and they said like, well, we know that in real life, nets are very complicated. They also have enzymes, and so in the last synchrotron run, we actually did experiments together with a largest group on human NCPs instead of xenopus NCPs. We also looked at what happens when you use these PAD enzymes, and we did a time-resolved experiments, and we showed that one of the really special things that you get with this is that you get a sort of time profile in the inflammation, because the enzymes will actually take away the positive charge in such a way that it will actually start turning this down after a finite amount of time. So you don't get this chronic inflammation that you do when you have a kind of out-of-control process, like sepsis, for example. So anyway, for this work, we again went to our old friend, Yuri Dobnikar, who came up with a kind of phenomenological theoretical description of this. And as you would expect, it's something that depends on the binding strength, which is something that we use to parameterize the matching between the DNA periodicity and the toll-like receptor 9, but also the domain size of the system. How many parallel strands of DNA are you presenting in order to get this immune response? And the more repeats you have, the greater the amplification that you have. So I think with this, I want to just end with some concluding comments, right? So what we're seeing here is a kind of deterministic control of immune activation, which is, kind of a holy grail for controlling the immune system. And we find that AMPs with a specific range of sizes and charge can lead to quantitatively distinct levels of immune activation. And what we're seeing is that these AMPs do a lot more than what they were previously thought to be their main function, which is to punch holes in bacterial membranes. We find that they actually assemble into these protofibrils that can organize immune ligands and give you this kind of systematic multivalent presentation that gives you immune amplification. But at the same time, you can also imagine anti-inflammatory pathways using peptides like the Theta defensins that can outcompete the pro-inflammatory guys and turn things down and give you results that are kind of like emblematic of what you saw with that rheumatoid arthritis mouse model. And we ended with a kind of exploratory project where we looked at what happens to the release chromatin how it can be remodeled into these columnar complexes in these so-called neutrophil extracellular traps and in necrotic cell death, giving you a kind of pro-inflammatory outcome. And the final take-home message really is just that these immune receptors, which are usually thought to be receptors that are sensitive to chemical patterns on individual molecules, turns out they're able to also sense crystalline patterns and not just molecular patterns. And this actually is because the ligand clustering can actually drive receptor clustering and give you these huge amounts of immune activation. So anyway, finally, I want to thank the people who've done the group. The work I talked about today was mostly done by Ernest Lee and some former group members like Nathan Schmidt and Fan Jin and so forth. I've had all kinds of great collaborators on this, including local collaborators from NTU, Lars Nordenschild's group, and so forth. Finally, I want to thank you for your attention.
Very interesting. So my question is the following. In DNA, when you condense it osmotically at very large osmotic pressure, it goes into an orthorhombic phase where it also has 4 neighbors. And the reason why it does that are the angular dependence of the interaction between molecules. And if you don't change the symmetry of the phase, it would be orientationally frustrated. Now here, I look at almost exactly the same case, I would think. So there must be some kind of an orientational interaction that is frustrated, and the frustration is alleviated when you change the symmetry locally from hexagonal to square lattice. Would that be the correct way to look at this?
I think that would be one way to look at it. I think the constituents you're starting out with have low enough symmetry that I'm guessing that one would, one will always want to make sense of it using the kinds of arguments that you just presented. But I would imagine the most general way of looking at it would be, some kind of Sharon Glotzer machine learning kind of approach where you look at how these things depend on small changes in parameters, like the amount of bending in the alpha helix, for example, will be important because we find that if the alpha helix is not bent, it will never do this, right? Also, how do these guys fit together to form to stabilize the right hydrophobic core and so forth? But these orientational interactions are going to be important. But what we do not know ahead of time from the qualitative arguments is whether you get like a large near degeneracy in low-lying states or something where, what we find is that when we did the simulations with Peng-Yu Ren, or rather when he did the simulations and told us what he got, is that when we flipped the polarity of the fibrils, it didn't seem to make too much difference in the inter-DNA spacing. And it was, so at that level, it didn't matter as much. But I think if you look at the most general case, everything that you said has got to be important.
As we limit the questions to just one more question and then talk during the break. I think you were.
Yeah, very nice, very convincing. Nice story about disease and structure. Let me ask you a question that you must get asked a lot. When we have the actual disease, we are in a biological medium. Proteins, ionic strength, whatever, hundreds of components. And here we have the components that you can look at by synchrotron or by whatever means. And we all know that structure changes even by the concentration of the components in the polyelectrolyte complex. How relevant are actually these in vitro or these structural studies with respect to what is occurring in the biological reality?
I think I can maybe, in the interest of time, answer that in 3 short different ways, right? One, we try to use solution conditions that are relevant in biology. We don't use distilled water, we use like 150 millimolar salt and so forth. And what we find is that it changes the spacing slightly when we did a phase diagram where we systematically changed the amount of monovalent salt and it didn't seem to matter. We also used serum, which is actually a good way of doing a kind of sanity check. serum has like a ton of different components besides salt, and we find that these structures persist in serum. The peaks are a little bit weaker, but you can still see the same periodicities. And finally, we did this. We generate these complexes, and we actually feed them to human plasmacytoid dendritic cells that are harvested from donors in Switzerland, and we showed that they activate those cells, right? So I think you never know for sure until you do stuff in animals, which we are starting to do, but it's a good question.
No, actually, I would like to put it a little bit further. For example, we have been looking at cation and DNA interaction, and we see that we get from a scrambled egg random structure in the polymer particle, we go to a core shell structure as a function of the concentration of things. And I think there are fundamental, even with the concentration of the components, you may not actually be sure. So these that you are looking at, they are stable in serum, and they can actually, there are structural similarities. But for example, the synchrotron, do you do it at the same concentrations as they occur normally, or is that
No, at synchrotrons, you usually do it at a higher concentration.
Yeah,
but I want to turn your question around a little bit, because it actually, it's a really interesting question, because what it shows is that it's yet another avenue for biology to turn things on and off immunologically, right? You already see that with these enzymes, I didn't show all the data, but with the enzymes, you're already attenuating these structures, and you are already able to actually start shutting down the immune response, which absolutely needs to be done in a real-life biological system. Otherwise, you have these chronic inflammation diseases that you see here, right? So the fact that these structures are, can be labile is actually a strength and like a strong feature of this model, rather than like a potential limitation.